Hello everyone, welcome back. So today we'll be doing the basic things about chromatin. Okay, so we have already completed nucleus, we have seen nucleus structure. Then I had told that chromatin will be doing later. And what is the main theme of today's lecture is that we are going to understand completely the basics of all the thing about chromosome, DNA and chromatin as well. Okay, because what actually happens, what I have observed is that many students, they are completely confused in these things, like what is chromosome, what is chromatin. Now usually we find DNA in the nucleus, then they say chromosome is there in the nucleus, they say chromatin is there in the nucleus. So students are literally confused about these things. So what I am thinking is that however these things are actually there for molecular basis of inheritance chapter, there we will be seeing all the thing about DNA and everything, but it is very much important to understand these concepts now also, because however we are going to study after this cell cycle, so there it is again important. So I thought I should completely clear it so that it will be easy for you for the next things. However, it should be clear today only. Okay, so yesterday we saw that this was the nucleus. We saw this was the nucleus, and then inside the nucleus we had this nucleolus, right? And then what we saw that there was a thread like material which was there inside the nucleus, and we named it as what? We named it as chromatin. What do we named it as? We named it as chromatin. And today's class we are going to start about what? We are going to start regarding chromatin. Basics of chromatin, you can say, the basic of chromosome, you can say. Okay, structure of chromosome, functions, all those things, the types also will be studying in the next class. But now we will be understanding what? Chromatin and all the things related to chromatin and other things. Okay, so let's begin then. So here inside the nucleus we saw that there is a thread like material called chromatin. Now what is chromatin? Okay, see, I'll try to explain it in the most simple manner, okay, so that you will understand it better. Now what is chromatin? This is the question. Then we know that inside the nucleus what will be present actually? It will be the DNA. So then what is chromatin and what is DNA? See, the DNA which is present in the cell, it is about 2 meters in length. See, every organism, every living organism, the cell of living organism, it will have this DNA, its own DNA. Now, the thing about DNA is that the length of DNA, here if you see, it is about 2 meters length, and we need to put this DNA, or we need to, what you can say, put this DNA into the cell, and that too we need to fit it inside the nucleus. So if this is the cell, inside the nucleus we need to fit the DNA. Adralenappa dod vishaya. But if you see, it is 2 meters long. Can you imagine if I give you a thread which is of 2 meters long, and if I tell you to put into a structure which is very, very tiny structure, then what will you do? What we need to do? Obviously, to fit it into that particular structure, that tiny structure, we need to what? We need to coil it, right? We need to coil it. If you coil it, then it becomes a tiny thing, then you can put into that. So that is how what we need to do here is, the DNA which is present in our body, this DNA is 2 meters long. So what we need to do? We need to coil it. How will you coil it now? That is the question again. So how we are going to coil it? How we will coil it? So when we need to coil the DNA, there should be something added, okay, so that it holds the DNA after coiling. Now what can we add here? Now this is the DNA. What we will do is, we will add some proteins to it. When we add some proteins to this DNA, these proteins, these are nothing but proteins, and these proteins are called histone proteins. What these are called as? Histone proteins. So why we are adding this DNA, this histone protein now? The basic idea is that to coil the DNA which is too long, we are adding some glue type material to coil it, and that is nothing but histone protein. Now the peculiar thing about histone is that histone is positively charged. Histone is positively charged molecule, or you can say positively charged protein. And what about DNA? Obviously, if histone is getting attached to DNA, then the DNA is obviously what? DNA is negatively charged. Yes? Are you understanding this? Why DNA is negatively charged? DNA is negatively charged because it has phosphoric acid in it. It is a nucleic acid. It has a phosphoric acid. Because of that, DNA is negatively charged. In order to coil that DNA, we need to add something which is positively charged so that it will coil the DNA. It will wrap around it. So that positively charged material is histone. So now, what is chromatin then? Actually, we are talking about chromatin, no? Now, from where these all things came? Yes, we are talking about chromatin and we are talking about DNA because DNA along with this histone protein is called as chromatid. Is this clear now? What did I say? What I said just now. DNA along with histone is called chromatid. So if you need to study about chromatin, then we are studying about what? We are studying about DNA. Not only DNA, we are also studying about some proteins. There are so many other proteins also. Non-histone proteins are also there. But main focus is on what? Main focus is on histone protein. Because this is a protein which is making that DNA condensed or coil. I hope this much is clear. Now what is chromatin? Did you get an idea? Whenever you come across this term chromatin, remember chromatin is nothing but DNA plus what? Protein. Right? Now which protein? It is histone. This thing you need to remember. Histone is positively charged, DNA is negatively charged. Clear? Please note it down. Okay, I hope this much is clear. So what is chromatin? Chromatin is nothing but DNA plus histone. And why this DNA is associated with histone? Because DNA is so long, we need to coil it and put it into a small structure called nucleus. We have this histone with it. So that histone will coil the DNA. It will make that DNA wrap around itself. We will see how it will do that. Okay, please take down this much. So we have understood what is chromatin now. Now usually what will happen is that, now let us consider this is histone protein. These are histone proteins. Okay, now let us consider this is DNA like this. Okay, like this, and these are what? The proteins which are associated with this. These are called histone proteins. Right? So what will happen if histone is present along with DNA? It will make this DNA wrap around it. So you will see that because histone is positively charged, what will happen? DNA will be wrapped around this histone protein. Are you observing this? What is happening here? This red colored thing, what I have written? It was histone. And what is this blue matter here? The thread like thing. It's nothing but the DNA. Now if you see this DNA properly, if you see this DNA properly, how it looks? It will just look like this. If you zoom it out, it looks like this. But I am showing it in the form of thread because that is how we are going to study there. This is nothing but the same DNA. So DNA is wrapped around what? It's wrapped around histone. So this is what? This structure is chromatin. I hope this is completely clear now. Chromatin is DNA and histone. Why histone? To coil the DNA. Now the next thing. Now when this cell is in this stage, now for example, let us consider this is a normal stage of a cell. Cell is under the stage called, this stage is called interphase. This stage is called interphase. That means cell actually metabolically it is active. It is going to, next level is division. It is going to divide now. Cell is about to divide. Now when cell needs to divide, you know that cell divides in such a condition. This is one cell. It is having its own nucleus, its own DNA. Okay. Now it needs to divide. What will happen? It will divide. It will form two daughter cells. The nucleus also will divide into two. The DNA also needs to be what? It will be divided into two and it needs to be equally put into these two daughter cells. Am I right? Cell when it divides, nucleus also divides, the DNA matter also divides. It needs to be divided equally and it needs to be put equally into the two daughter cells which it has produced. Right? Now when this thing should happen, okay, when this thing should happen, what can you understand here? Now when you're equally dividing the DNA, is it easy for you to divide DNA if DNA is in this condition? You can see that DNA is just like a thread matter. Now can you equally divide it? If you divide it like this, for example, is it equal? Do you think it is equal now? It is having less, it is having more DNA. So this cannot be done. This cannot happen. Then what we need to do? We need to make some structure out of this DNA so that we can equally divide it. So what can be done? That we will see now. So here what happens to this structure? This is chromatin now. So this is chromatin. This structure is what? It is chromatin. Now what will happen to this chromatin later on when the cell starts, it wants to divide? During that time, this chromatin starts condensing more. There will be more condensation happening during that time when cell is about to divide. Now how that more condensation will happen? See, what will happen is that there are some structures here, these histones. If I am talking about histones, there are some structures called histone octamers. 4 histones, behind there are 4 histones. So totally there are 8 histone structures. 8 histone structures, and this 8 histone structure we call it as histone octamer. Okay, now see, histone is positively charged, DNA is negatively charged. Now for one histone, so much of DNA is wrapped. Now you imagine there are 8 histones. Then how much DNA will be wrapped here? If I talk about DNA, in this histone octamer, DNA will be more. DNA will be wrapped around it. Are you understanding this? These things are what? These things are histone octamer. Correct? Correct alva? Idu histone octamer alva? Because 8 histones, 4 at the front, 4 in the back. 8 histones are there, which is called histone octamer. For one histone, so much of DNA is coiled. Then if there are 8 histones, then DNA amount which is coiled will be more. This is DNA. Correct alva? Now this structure is called nucleosome. What it is called? It is called nucleosome. So I'll rub this. It is histone octamer. It is made up of 8 histone and this is DNA. So now histone octamer plus DNA, it is called nucleosome. That is why in case of molecular basis of inheritance chapter, you will study packaging of DNA. There you will be studying this nucleosome model. Okay, but why I am explaining here, because you need to understand this is it's completely a basic thing. Now this is called nucleosome. Such type of nucleosome will be there in the what? During the cell division process, when it is going to take place, that time histone octamers will come, and what they do is they start coiling this DNA more. Are you understanding? Now what will happen? Because of histone octamer, so much of DNA will start coiling around this, like this. So much of DNA is getting coiled around it, like this. So what is happening? DNA is getting more and more condensed. Its complete condensation of DNA will take place. Why it is happening? Because cell is going to divide. What we need to do? We need to divide the DNA also properly. So finally what will happen to this is that, so I hope this much is clear now. So this is about nucleosome. So many nucleosomes come together. They come together. They are coming together. Why? Because they want to condense the DNA and make some particular structure. We will see that. What will happen is that, totally if I show you, after so much of condensation, if this is DNA like this, I'll show it with black. If this is DNA like this, now it starts condensing like this. So now you can see, yeah, there is a particular structure. So much of DNA was there. Okay, now equally they have been, they are converted into what? This structure. Now this structure, if you see, it has a central region here, and this is called chromosome. This is called chromosome. I hope you people understood. So many students, they don't have this knowledge about whether DNA has chromosome. Chromosome is made up of DNA. A lot of confusion students will have. So it is clear now. DNA plus protein becomes chromatin. Chromatin will condense to form chromosome. So this only more condensation will happen to form this chromosome. Now then, okay, these type of structures, now after interphase, when it comes to the next phase of cell division, actually don't worry about these stages. We'll be doing it in the cell division chapter, but still you need to know it. So next stage will be what? Will be prophase, which is the first stage of cell division. During prophase, you will find these type of structures in the chromatin. The chromatin will move out. Chromatin has got condensed completely. Slowly what will happen? Slowly nucleus also will be gone. Nucleus also will be gone. And what is remaining here? What will be remaining here is the condensed chromatin. What is that condensed chromatin now? This only, no? Condensed chromatin, I'll show it with blue. Okay, right. Okay, this condensed chromatin, what you find here? These are what? These are chromosomes like this. How many? If it is a human cell, how many will be there? If it is a human cell, such type of chromosomes will be what? 46 chromosomes. How many? 46 chromosomes will be there in a cell. 23 pair. 23 from father, 23 from mother. If it is mitosis, then what will happen? The cell divides and forms two cells. Again, both the cells will have 46 and 46. If that is the condition, you understood this one. This is chromosome. These are chromosomes. I have not shown particular number just to show you. It's not 23 or 46 here. Just a number. Okay, 6 I have done. Now, in case of mitosis, it is equational division. There in mitosis, what we need to do is, we need to divide the cell equally. Chromosome number should be equal in both the daughter cells. For example, how many are there here? 6 chromosomes are there. Now, this cell will undergo mitosis, for example. I'm just showing you, for example, if it will undergo mitosis, what will happen to this now? It needs to 6 chromosomes should be there in this also, 6 in the other daughter cell. Both the cells should have same number of chromosome. Already it is having 6. How can it provide 6 and 6 for both the daughter cells? Usually division means what? Agar 6 hai, 6 ide andre, if you are going to make 2 cells out of it, we think it is 3 will go here, 3 will go here. Yeah, that also is there. That kind of division is called meiosis, wherein it is halved. But this is not meiosis, this is mitosis, where we are giving equal number of chromosomes. That means, first of all, you need to make it double here only before division. Means you are going to make what? You are going to make it replica. Same thing should be there, no? It should not be different here. Same chromosome should be there. both the side. I mean 12 chromosome totally. How 12 chromosome will be formed? What will happen then? See here, what happens is this particular chromosome will replicate. I mean the DNA will replicate, and then it forms the other form of this before division, like this. Are you understanding what I am trying to say? It will form the replicated part like this. Now I think your doubt is cleared. You might be thinking, no, whenever we have seen chromosome, this is the structure we have seen, and ma'am is showing this type of structure. I think it's now clear for you. When it is undergoing mitosis, it needs to divide in such a way. I mean, now what will happen is that one part, if you see this part is equal and identical to this part. This chromosome and this, both are chromosome only. Xerox copies of each other. Clear? Both are Xerox of each other. Now in mitosis what will happen? The center will be cut, so that this part will go to this, the other part will go to this. Same thing is moving both the side. I think you understood what is chromosome. We are going to talk about each of these stages in the next chapter, cell cycle and cell division. I think you understood what is chromosome now. And why is this structure? Is this clear? Chromosome is made up of what? It is made up of DNA plus nucleosomes. Nucleosome means what? That is nothing but histone octamer plus DNA. Histone octamer is what? 8 histones joining together. What are histones? They are the positively charged proteins which help in coiling the DNA, also which help in condensing the DNA into chromosomes. So before cell division, when cell is in the normal stage, it will have a thread-like material called chromatin. When cell is about to divide, the chromatin gets condensed to form this chromosome. And later on, cell will divide, and these two daughter cells also, after division, they also will undergo again interphase. Again this chromosome, it will decondense. It will decondense, and then it will form what? Again it will form chromatin. So it has this power of condensation and decondensation, you can say. Okay, that we will see in the next this one. I hope this much is clear now. Okay, so this is, please, if you want, you can just take that. Okay, so what is chromatin? DNA and histone. I think it is clear. Okay. Okay, now if you see this chromosome, okay, if you see this chromosome is what? This chromosome, you will be able to see this chromosome only when cell has crossed prophase and it is going for metaphase. During metaphase, you are going to observe this kind of chromosome. This type of structures you will see. And the thing is that how they are joined together, how they are together, because there is a central region called centromere, and this centromere will hold this chromatid and this chromatid. Now, okay, I did not tell you what is chromatid now. See, chromatin condensed to form this chromosome, right? So this is one chromosome completely. And this chromosome, now if you see this normal chromosome like this, it will have this centromere, and this part, we call these two as chromatids. We call them chromatids. This is one chromatid, this is one chromatid. Arms of chromosome, we call it as chromatids. This is one chromatid, this is one chromatid. Now, so this is chromatid, this is also chromatid. Now they have their replicas also, which are equal and opposite, not opposite, equally, they are equal, Xerox copy of each other. This chromatid and this chromatid are equal, they are same. So these two, we call them sister chromatids. I'm talking about the whole thing, okay? So this is one chromatid, this is one chromatid. Both are equal. So we call them sister chromatids. This is one chromatid, this is one, and both are what? Sister chromatids. So when you consider here, okay, in case of human beings, we have 46. Okay, let it be, we will talk about that in next class. Here, I think this much is clear. Chromosome will have centromere region where it will hold this chromatid properly, and the centromere will also hold the other chromatid also. So in a single chromosome, if you see, we will call it as chromosome only, because there is no different chromatids here. Chromatids are all equal, same. So this chromosome structure is having chromatid, and this chromatid is sister chromatids. Understood now? Now if any other chromosome is there along with this, then this chromatid and that chromatid will be non-sister chromatids. They are not sister chromatids, they are non-sister. If you are considering them with this chromosome with another chromosome, then those are non-sister chromatids. That we will be talking in detail in the next video. Okay, I think this much is clear. This basic knowledge is very much important, so I thought I should clear this basic first before going next for the next video. Next we'll be seeing this structure of chromosome properly, exactly what are the parts, okay, and how it functions. And in NCERT particularly, they have mentioned about different types of chromosomes. That also we'll be seeing. So please watch this video how many times you want, but you should be, the knowledge whatever I have taught today, it should be fixed in your mind, so that next video I will not tell everything again and again. I'll be continuing with the terms chromosome, chromatids, sister chromatids, non-sister chromatids. So you people should be aware about that. I hope you will. Okay. Okay. Thank you.